Welcome everyone. Today we're going to shrink ourselves down and go on a safari. But forget the Serengeti, we're heading somewhere way more crowded and complex, inside a single living cell. You can think of it as a bustling, microscopic city, and we're about to take a tour of its most amazing machinery.
So here's our game plan for the trip. First up, we'll check out the cell's moving parts. Then we'll pop over to the central organization hub. After that, we're going deep inside the command center, unpacking the genetic blueprints and looking at some tiny, specialized micromachines before we wrap it all up. Sound good? Let's go.
Okay, our very first stop is right on the cell surface. This is where we find the structures that get everything moving, the cilia and the flagella.
So what exactly are these things? Well, cilia and flagella are basically the cell's propellers. They're these little hair-like things that stick out and whip around, working just like oars on a rowboat. They really have two main jobs. Either they propel the entire cell through liquid, or the cell stays put and they sweep that liquid past the surface.
Now, if you could slice one of these little oars open and peek inside, this is what you'd see. The core, which has the technical name axoneme, isn't just a simple thread. As you can see from this diagram, it's an incredibly organized, complex piece of machinery. This precise structure is the secret to how it generates so much force.
And this whole intricate engine is built on this really elegant repeating pattern. Biologists call it the 9+2 array, and this signature arrangement is the key, the absolute blueprint for how every single one of these things works.
Okay, so let's break down this 9+2 array. Around the outside, you have 9 pairs of tiny protein tubes, we call those microtubules, that form the outer ring. Those are the peripheral doublets. Then, smack dab in the middle, you've got 2 more single microtubules. The radial spokes connect that outer ring to the center, kind of like spokes on a wheel. And these inner doublet bridges link all the outer pairs together. This entire system of rods and connectors is what lets the whole thing bend and create that powerful whip-like motion.
So we've seen the motors, but where in the world are they built? For our next stop, we have to travel inside the cell to what I like to call the construction site for these structures, the organization center or the centrosome.
The centrosome is a really fascinating little thing. Its most important feature are these 2 cylinder-shaped structures called centrioles. And what's really distinct about them is their arrangement. They're always lying perpendicular to each other in an L shape. And believe me, that orientation is absolutely critical for their job.
If you took a cross-section of just one of those centrioles, you'd see this incredible cartwheel structure. It's made of 9 evenly spaced sets of protein fibers. This design gives it amazing strength and organizational power. It's just a perfect example of form perfectly matching function.
So what does this construction site actually do? Well, it has 2 absolutely critical jobs. First, it forms what are called basal bodies. You can think of these as the concrete foundations that anchor the cilia and flagella we just talked about to the cell. And second, in animal cells, the centrosome builds the spindle fibers. These are like tiny ropes that pull all the genetic material apart when the cell divides. So yeah, it's essential for both movement and reproduction.
All right, our safari has now taken us to the main event, the real heart of our microscopic city. Let's head inside the command center, the high-security vault where all the blueprints are stored, the nucleus.
And here it is. This image gives us a fantastic view inside the nucleus. You can clearly see it's a completely separate compartment, and inside is where the cell protects its most valuable asset, its entire library of genetic information, the DNA. Protecting that information is priority number 1, which is why the nucleus is built like a fortress. It's surrounded by a nuclear envelope, which is actually a double membrane, a wall within a wall. The gap between those 2 layers is called the perinuclear space. But a fortress needs gates, right? And that's what the nuclear pores are. They're like carefully guarded checkpoints that control exactly what gets in and out.
So what's actually in this vault? Well, when the cell is just going about its day-to-day business, the DNA isn't all neatly bundled up. Instead, it's a tangled network of DNA and proteins called chromatin. The best way to think about it is like a library where all the instruction manuals are just lying around in a big pile, ready to be grabbed and read at any time.
But when it's time for the cell to divide, you can't have a messy library. Everything needs to be organized and copied perfectly. And this is when the chromatin undergoes an amazing transformation, packing itself down into these dense, manageable structures we can actually see with a microscope, the chromosomes.
This is what a fully packed chromosome looks like, all ready for cell division. On the left, you see that classic X shape we're all familiar with. But, as you can see on the right, they don't all look the same.
Chromosomes actually come in a few different shapes, and it all depends on where that pinched-in middle section is located.
So let's quickly break down the anatomy. That X is made of two identical sister strands, called chromatids. They're held together at that pinched-in waist, which is called the centromere. Now, sitting on the centromere are these little disc-shaped structures called kinetochores. And these are the critical docking points where those spindle fibers we talked about grab on to pull the chromatids apart.
The location of that centromere is what gives each chromosome its specific shape and its name. This table makes it super clear. If the centromere is right in the middle, creating two equal arms, we call it metacentric. If it's a little off-center, making one arm short and one long, that's submetacentric. When it's really close to one end, you get an acrocentric chromosome. And if it's right at the very tip, it's teleocentric, leaving it with just one arm.
For the final stop on our safari, we're going to zoom in on some of the cell's smallest workers. After seeing the big command center and the library, you can think of these as the specialized utility closets of the cell, known as microbodies.
Now, these are pretty simple, but they are incredibly important. A microbody is really just a small, membrane-bound sac. And what's inside? Enzymes. Each type of microbody is packed with a specific set of enzymes to carry out a very particular metabolic job, which keeps some specialized and sometimes pretty dangerous chemical reactions safely contained.
And that brings our safari to a close. We have seen some absolutely mind-blowing microscopic machinery today. So, let's do a quick recap of our journey just to lock in everything we've seen.
This table really puts it all together. We started with cilia and flagella, with their 9+2 axoneme powering movement. Then came the centrosome, with its perpendicular centrioles handling organization and division. We went inside the nucleus, that double membrane fortress that protects our DNA. We saw how that DNA gets packed into chromosomes, with their key centromeres. And finally, the simple but essential microbodies, those little enzyme-filled sacs for specialized reactions.
Before we go, I want to leave you with this one fact that is just unbelievable. Every single human cell in your body contains about 2 meters of DNA. That's over 6 feet, and it's all packed into a nucleus that's just a few micrometers wide. That is an absolutely incredible feat of engineering, and it's happening inside trillions of your cells this very second.
And that leads me to a final thought. All of this amazing machinery, from the motors to the command center, is built from a single set of instructions. So here's a question to think about. How can tiny, almost insignificant errors in copying that code lead to such massive, life-altering consequences for the entire organism? Thanks for joining me on this explainer.